This is Dr. Wu. My lab is working on computer-aided drug design. Let me first tell you why we want to use computer for the drug discovery. The traditional way is an experimental-based approach, which is cost a lot of money and many years to develop a new drug. The fundamental reason is a blind testing. And typically it will start from the large number of the compound. So you have to synthesize, purify it, and then you move it into the pipeline testing, preclinical trial, clinical trial, and hopefully you have compound able to approved by the FDA. But you see, spending a lot of the time, resources on bad compound. The rationale to do the virtual screening is, well, we know a lot of information about the drug target. Typically, it's a protein. We know its sequence. We know its structure. We know its active site. So we can model the interaction between the drug candidate with the drug target. So then we can do the virtual screening in the computer, which are faster and cost penny. And so in this picture, you see my student basically sit in front of the computer to use software to model the interaction between the drug target and the drug. Okay, and besides molecular modeling, we also have another powerful tool to study the interaction between the drug and drug target. So we have allocation in the Fetish supercomputer for doing the molecular dynamic simulation. And then we can get insight about the interaction between the drug and the drug target. So for example, and this is actually a cancer drug, first line cancer drug for leukemia. And so we simulated its interaction with its target, which are the 2-strand DNA. And then the drug is supposed to be intercalate between the base. So we want to understand the detail how this drug able to insert. And you can see it started random search. And it had interacting with the DNA. And then the base pairing is open. So drug actually can insert between the base pair. And then you see the first layer of the base pair come back and form the hydrogen bond. Now drug bonds tightly between the base pair. So through those molecular insight can help us to optimize the drug to understanding how the drug work. The current we have working on the 3 major areas. So first area is the anti-cancer drug. We focus on discover the compound which are able to stabilize some of the abnormal DNA structure, which called the quadruplets. And once those quadruplets develop, it will interference the genetic information transcription and translation. So that will stop the cancer cell cycle. And here's a simulation show you how the compound able to stabilize the G quadruplets abnormal structure and interface with the duplex. And this provides us a lot of the detailed information about the interaction. The second area is the anti-COVID-19 drug. So we have collaborated with several experimental groups to do the virtual screening, identify the good candidate for various of the viral target, including the target which has the first FDA-approved drug, which is the RNA-dependent polymerase, and remdesivir is the first FDA-approved drug. So we're looking for some of the new derivative of the remdesivir. We're also looking for some of the more potent compounds might be good replacement for the remdesivir. The third area we're working is the membrane receptor area, which are linked to the neurologic disorder like the pain and antidepressant and Parkinson's diseases, schizophrenia. And here what I show you is we try to develop some of the partial bias agonist targeting dopamine 2 receptor, which are important in some neuron disorder. Okay, so you're welcome to do volunteer research in my lab, or you're welcome to do the research for credit, and we also have a paid position, and after you got the necessary training. And last year, our group published 11 papers, and this year, so far, we have already published 4 publications. So we're looking forward to your joining our team. And if you want to find more information about my research team and my research program, you're welcome to looking at our group website.